Welcome to Biology My Passion. I am Soumya Harikrishna. We are learning the chapter Biomolecules. We dealt with the analysis of molecules. We also learned biomicromolecules and biomacromolecules. Now we are coming to the last part of this lesson. It is about enzymes. During enzymatic reaction, first enzyme, it combines with the substrate at the substrate binding site and it forms enzyme substrate complex. Now, when the enzyme is bound to the substrate itself, this will make the change. Enzyme undergoes itself a change to fit into the substrate if it is not like lock and key. Lock and key means it correctly fits, or it induced fit model is there where enzyme will alter its shape to bind tightly to this substrate. Now, it is enzyme substrate complex. It will change or alter this chemical bonds in this enzyme substrate to make it into product. So what do we get? Enzyme product complex, and after that product will be released. So this can be asked as a 5-mark question. Enzyme action. The steps are given 1, 2, 3, 4, 5 in the textbook itself. First, enzyme binds with a suitable substrate at the active site or the substrate binding site. Second, once they combine, that is called an enzyme substrate complex, or enzyme alter shapes to fit the substrate. Third, enzyme will alter changes in the product, chemical bonds in the substrate to make it into product. So we get enzyme product complex. Then the product will dissociate from the enzyme and it is released out, and the enzyme is ready for another round of catalytic action. Let's now discuss different factors affecting enzyme action. First one is temperature. We already told temperature plays a significant role in the rate of enzymatic activities. And usually in animals, the optimum temperature is between 30 to 40 degrees Celsius, whereas in plants it is between 20 to 30 degrees Celsius. But there are thermostable enzymes in bacteria living in hot thermal vents also. Let's come back to the optimum temperature. So the enzyme action is maximum at an optimum temperature. Above that and below that, the temperature will disrupt the enzyme action. How? Because if the temperature is going above 45 degree in a normal conditions like ours, what the problem is, the enzyme get denatured because its structure will be disrupted. The denaturation, the typical example we can remember is the egg. Egg white is albumin protein. As soon as we boil it or the temperature increases, it solidifies. Can you bring it back to the liquid form? No. This is called a denaturation of the protein. The same way, if the temperature is low, it will not disrupt the structure, but it makes the enzyme inactive. That is the principle that we use in refrigeration. We are keeping the things like a meat, fish, etc. in the freezer. Why? We are suspending the enzymatic activities so that the bacterial activities will not be there at that suspended or low temperature conditions. Understood? So we know that when we increase the temperature, initially the rate of reaction will increase, but only up to a limit. After that, the reaction rate will decline. So if it is high temperature, it denatures the enzyme, or if it is low temperature, it inactivates the enzyme. Okay, so these two are the conditions. So we get a graph like this. Now next is the pH, the same. pH usually in our cell, most of the enzymes work at a neutral pH, but we know in the digestive system in the stomach, it is HCl present, so it is activated, or pepsin is activated at the acidic acidic pH. But when it comes to the small intestine, we know pancreatic enzymes are acting in alkaline pH. So bile is converting it into alkaline medium. So there the temperature, what is changing? The pH is changing. So it is again pH also, optimum pH it will be working. Below and above the rate is less. So this is the highest with the optimum pH. This is optimum pH, this is optimum temperature. Now next is a substrate concentration. Suppose you imagine one substrate will be adding to one enzyme. Suppose this is substrate, enzyme. So enzyme will bind to the substrate. Okay. Or I will give you an example. There are 20 chessboards I have kept. Okay. So 20 of you are already sitting on one side of the each chessboard. So I have 20 students. I need another 20 students to play with them. So first 5 students come. Okay, I allowed them. Another 10 students come, I allowed them. Then again 5 students come, I allowed them the places. So now all the 20 are saturated. Now another 5 students are coming and asking, ma'am, can we also play? Though I want to let them play, but there is no chessboard available because one chessboard can be used by 2 people. So it is completely occupied, right? The same way. Suppose 100 enzymes are there in the beginning. So I am adding only 25 substrate molecules. So the 25 enzymes will be taking them up. But still enzymes are available. So I increase the substrate concentration another 50 more. Then again it will increase because more enzymes can bind with that. Then again I am giving another 25. So the enzymes are all saturated. Now if I add more substrate, another 50, it will not affect the rate because there is no free enzyme to increase the rate. Okay, so the graph goes like this. It goes first high. This reaches the maximum because all the 100 are saturated. This maximum is called V max. That is, velocity of the enzyme is maximum at this level. After that it stagnates or it becomes steady position. So these 3 graphs and the factors influencing enzyme are very important. Just now we learned the factors affecting enzyme action. But apart from these, some presence of some chemicals or substances can also inhibit the reactions of enzyme or enzyme activity. They are called inhibitors. The substances which or chemicals which can inhibit the enzyme action are called inhibitors. Inhibitors can be of 2 types: competitive inhibitor and a non-competitive inhibitor. Competitive inhibitor means it resembles its shape similar to that of the substrate. So what happens, instead of the actual substrate, the competitive inhibitor will bind with the active binding site. For example, suppose we have an enzyme like this with active site like this, and the actual substrate has a structure like this. So usually what happens, the actual substrate will come and bind at this position, forming our enzyme substrate complex and continues the reaction. But here we have a villain in this picture. So this villain is having a shape like this. So this is the competitive inhibitor because its a binding site is similar. So what happens, instead of this inhibitor will come and bind here. So now the reaction cannot proceed as the substrate is not allowed to bind here because already another inhibitor has occupied that active site. So this is called a competitive inhibition, where the inhibitor structure resembles that of the substrate. For example, succinate or succinic acid is actually hydrolyzed by another enzyme called as succinate dehydrogenase. But this enzyme or the substrate succinate has a competitive inhibitor, malonic acid. So malonic acid resembles that of succinic acid. So it will bind with the succinate dehydrogenase and thereby inhibiting the reaction. Now second is a non-competitive inhibitor. For example, here again the enzyme is like this, okay, and our substrate has this structure. So the substrate is coming and binding here, but there is some other molecule which is coming and binding at a different position of the enzyme, and this is called a non-competitive inhibitor. But once this binds, our enzyme action will not happen. So this is called a non-competitive. Structurally it is not competing with the substrate, but its presence or its attachment to the enzyme at a different position can alter the enzyme activity. Let's now talk about the classification of various enzymes. The classification is done by International Union of Biochemistry and Molecular Biology. So according to that, there are 6 classes of enzymes depending upon the type of reaction they catalyze. First is oxidoreductase or dehydrogenase. You already learned about oxidation and reduction reaction in class 10. If oxygen is added or hydrogen is removed, that is oxidation. or if oxygen is removed or hydrogen is added, that is called a reduction. But in terms of electron also we can say, if electron is added, that is reduction, electron is removed, that is oxidation. So here oxidoreductase enzymes catalyze oxidation and reduction reaction. For example, here S and S dash are the substrates. S is the reduced and S dash is the oxidized. But this enzyme will catalyze the reaction in such a way that in the product S is oxidized and S dash is reduced. So both the reactions are being catalyzed by this enzyme. The second class of enzyme is a transferase. Transferase is actually transferring a group from one substrate to the other than hydrogen. For example, here S has a G group associated with it and S dash is another substrate. But in the product, the group G has been shifted to S dash. Now S is free. So this is called a transferase enzyme. Another one is called a hydrolysis enzyme. They actually do the function of breaking of large molecules into smaller ones by water molecules. That is, amylase is an enzyme. You know, salivary amylase in our mouth can convert starch into glucose, or sucrase is an enzyme which can convert sucrose into glucose, or lactase enzyme can convert lactose into glucose and galactose. Now next is liases. Liases and the last class is ligase. They have opposite meaning. Liase means breaking, ligase means joining. These are molecular glues. These are catalyzing the reaction by removing removing or breaking the covalent bonds and adding making the double bond. Okay, for example, here to see, first to see there is an X, see here also Y group. So this is satisfying the valency. Now these two are removed and made into separate group so that there is a double bond forming between carbon. So this is called a liase. It is lysing, means breaking that covalent bond. Whereas when it comes to ligase, it is joining the different molecules together. For this, they use the energy from the hydrolysis of ATP. For example, joining carbon and oxygen, or carbon and sulfur, carbon and nitrogen, or phosphorus and oxygen. Third is isomerase enzymes. They catalyze isomerization reaction. Isomers, you know, they have same structural formula, same molecular formula but different structural formula. So they make changes in the structure to form isomers. So this is very important, 6 classes of enzymes. The last part discusses about cofactors. We learned that all enzymes are proteins. So suppose this is the protein part of the enzyme, that is called a apoenzyme. Okay, so this is the apoenzyme or the protein part of the enzyme. But this enzyme is not active until and unless there is a non-protein part associated with this. This can be called as a cofactor. So the cofactor binds with the apoenzyme to make it active. They are called a cofactor. So now it is called a holoenzyme or the active enzyme. Okay. So apoenzyme is the protein part, cofactor is the non-protein part associated, but they are inevitable for the action of enzymes. So, but these cofactors can be of 3 types: prosthetic group, coenzyme, or metal ion. If this cofactor is bound to the enzyme tightly and cannot be separated, enzyme means the protein part, apoenzyme tightly, and cannot be separated, then it is called a prosthetic group. For example, there is an enzyme called a peroxidase and catalase for conversion of hydrogen peroxide to water and oxygen. For this enzyme, there is a heme group. Heme means iron group, tightly bound to that apoenzyme always. That is called a prosthetic group. Now, if this cofactor is a transient association, but it can be removed, that is called a coenzyme. For example, the nicotinamide adenine dinucleotide NAD or nicotinamide adenine dinucleotide phosphate, these are the energy rich compounds formed in the photosynthesis. So these are having a transient association with the vitamin niacin. So that is only a transient or a temporary association. So that is called that cofactor is called a coenzyme. Okay. Then third, some of the enzymes need certain metals associated with them for their proper functioning. For example, carboxypeptidase is a protein digesting enzyme produced by our pancreas. So for that enzyme to act, we need zinc as the metal part. So that is another cofactor. Hope you understood all the concepts I discussed here. If so, please give a like and also share with all your friends. And if you haven't subscribed to my channel yet, please do so. Thank you for watching Biology My Passion.